Today we will be presenting about bacteriophages. Our group number is group 6 and our group members consist of Ashley Alonso, Juliana Bach, Michaela Larman, and Brenda Vela. Today we will be presenting an introduction, current knowledge, impactful research, and summary. My name is Brenda Vela and I will be introducing to you bacteriophages. So bacteriophages. Bacteriophages are viruses that infect and replicate in bacterial cells. Their morphology consists of a capsid, which is a rigid symmetrical container composed of the viral protein, a nucleocapsid, which is a capsid with the enclosed genome, a viron, which is the infection particle. And phages are usually not motile and depend on Brownian motion. We'll be talking about bacteriophages' species-specific nature, the replication consisting of the lytic cycle, and the replication consisting of the lysogenic cycle. Bacteriophages consist of a very species-specific nature. There are receptor bindings, which consist of the glycoproteins, which are proteins on the viral envelope with a sugar molecule attached. Surface protrusions, which are spike-like surfaces that allow the virus to latch. Cannula-like depressions, which are cannula-like depressions that act as binding sites for host cell receptors. They also have a species-specific nature due to their injection of the genome, which after binding the phage attaches to the bacterial surface. Then penetration happens where the phage injects DNA or RNA utilizing specialized cell structures, such as a tail sheath or a syringe-like apparatus. They also have genomic compatibility, as in their promoter recognition from the host RNA polymerase has to be recognized in order for transcription to occur. Phages can also have DNA or RNA as their genetic material. Single-stranded or double-stranded phages must first form a complementary strand before transcription occurs, while double-stranded phages have a full template initiating transcription. Replication in bacteriophages consists of two cycles, the lytic cycle and the lysogenic cycle. The lytic cycle consists of attachment penetration, replication transcription, assembly, lysis, and release. For the lysogenic cycle, um, it consists of attachment injection, integration, replication, induction, and lysis. Differences between the lytic and the lysogenic cycle is that the lytic cycle um, is lyse, which release newly formed phages um, which infect other cells. For the lysogenic cycle, the host is replicated within the cell and they are replicated with the host genome, the viral genome as prophage.
Hi, my name is Juliana Bach, and I will be speaking on the topic of our current knowledge of bacteriophage therapy. So, what is phage therapy? Phage therapy is the use of bacteriophages to treat infections. When people get sick, we had often treated them with antibiotics, and that worked well for a while. But now, the infections we have been treating have built up a tolerance to the antibiotics, meaning that the treatment is no longer effective. With increasing drug resistant bacteria, phage therapy offers an interesting alternative. Bacteriophages can target pathogenic bacteria and kill it. This treatment has potential in treating infection antibodies can't. A common issue with antibiotics is that they are not specialized, and it kills the infection along with other microbes in your body. But bacteriophage therapy targets specific pathogenic bacteria, and this treatment does not disrupt the human microbiome. So, In this slide, I'll be talking about the ways that phage therapy is innovative. So they are specific. Phages target only the harmful bacteria. This preserves the beneficial microbia in the body, which also helps reduce the side effects that antibiotics would have on a patient. They also have co-evolved. Phage and bacteria have evolved together for millions of years, and they have, and they will, and they are still doing it. So phages can adapt to bacterial resistance and mechanisms. This offers a dynamic treatment approach, which is very helpful for our ever-changing world of viruses, right? And they also have synergetic effects. Phages can be used in combination with antibiotics to enhance effectiveness and reduce resistance. This means that we can still use the medicines we have and we've already created, so nothing goes to waste, but they will improve on their effectiveness. According to the UN in 2019, they estimated that 1.2 million deaths worldwide were caused by drug-resistant infections, while 4.95 million deaths were linked to an antimicrobial resistant bacteria. So I found this chart that shows that antibiotic-resistant infection could kill 10 million people within the next 30 years. Though the threats are worldwide, low-income countries like Africa and Asia will be the most affected and are expected to have the highest death toll. Unless drastic measures are taken, it could push 24 million more people into extreme poverty and make the GDP fall 3.4 trillion annually in the next decade. Organizations like the World Health Organization have recognized the potential of phage therapy, and as a part of the effort to battle the antimicrobial resistance, the World Health Organization has called for an increased research into phage therapy as an alternate to traditional antibiotics. On the other hand, the National Institute of Health has been funding studies to explore the potential of bacteriophages in treating multi-drug resistant infections. They have even engineered phages to make the perfect medicine to fight infections. Next, I'll hand over the PowerPoint to Michaela, where she'll explain the impactful research of bacteriophages.
Hi, my name is Michaela Larman, and we will be discussing the impactful research that is being done about bacteriophage and 2 cases that without phage therapy 2 lives would have been lost. Also, how CRISPR has been significant in adding to the key research about bacteriophage. This slide is about why phage therapy is needed in the fight against MDR bacterial infections and why it is a threat to global health. Multi-drug resistant bacterial infections can be a threat to global health, reducing the effectiveness of antibiotics and increasing death rates throughout the whole world. Bacteriophage therapy, which uses the virus to specifically target and kill bacteria, has become an alternative treatment to aid patients that are in need of help. This case study explores explores how phage therapy is used to treat MDR infections, focusing on how it works, its results, and its future possibilities. This slide contains before and after pictures of a case of a 15-year-old who already had a pre-existing medical diagnosis and who unfortunately developed a drug-resistant infection called Mycobacterium abscessus. It was a groundbreaking research into this type of infection that gave the way to identify the 3-phage treatment that could specifically target the bacterial strain the patient had. Scientists, along with medical staff, engineered a phage sequence DNA that enhanced their ability to kill the bacteria to all 3 sites that was causing the resistance, so the antibiotics needed to help can be taken. This case is key because it demonstrates the effective potential of phage therapy and how this is a viable option for treating these infections. It highlights the adaptability of bacteriophage. This slide is about a case study involving Dr. Tom Patterson. He was traveling through Egypt on holiday when he became infected with an MDR infection, causing a fever that led him to slip into a coma. He was treated in Egypt and transferred to Germany, where he started to get better, but a turn for the worse came and he again declined in health due to an infection the medical team was still trying to determine. He was then transferred stateside for further evaluation that potentially saved his life. This case is key research because it demonstrates how phage therapy can be a necessary tool to combat antibiotic resistant infections. Scientists created a personalized treatment to safely administer intravenously to the infection site and target the specific bacterium. After close monitoring, the patient with the cocktail of phage therapy and antibiotics improved his health significantly. This last slide that introduces the impactful research that has been done in regards to bacteriophage is about CRISPR, which was first identified as a bacterial defense mechanism against bacteriophages. It plays a key role in phage therapy by using different sequences to target different bacteria. It can be used to engineer bacteriophage DNA sequences to specific bacterial infections that have become resistant to antibiotics, making treatments of these infections more efficient, safe, and with precision in combating the resistance. Doing this will help countless patients who can be battling many medical diseases and develop these infections and become helpless because of the body's resistance to what will save their life, which is why this is a major focus in the research of bacteriophage.
Overall, there are many positives that come with the potential bacteriophages possess. The bacteriophage can survive in plenty of tough environments like the human body. They diligently home in on their specific target and they annihilate it. So each phage is basically programmed to kill a certain bacteria, which they identify via the receptors that are on the surface of that bacteria. And that is where we get the idea and the theory that we could program these bacteriophages to target those specific bacteria that are antibiotic resistant. So usually when we think of bacteria infections, we're automatically thinking of treating them with an antibiotic. Now, usually for a severe infection, that is our only option. But due to many instances of antibiotic misuses, we're seeing an absolute surplus of superbacteria that are immune or resistant to our most powerful antibiotics. So if this trend follows suit, we're going to come to an instance where there's going to be a bacteria that's immune to every major antibiotic that we have. So this is where the study and advancement in clinical trials of the bacteriophages come in. We could save thousands of lives and start winning the war on bacteria if we use these bacteriophages that are available for us. Sadly, there are many factors putting a pause on this kind of advancement. Overall, there are many positives that come with the potential bacteriophages possess. The bacteriophage can survive in plenty of tough environments like the human body. They diligently home in on their specific target and they annihilate it. So each phage is basically programmed to kill a certain bacteria, which they identify via the receptors that are on the surface of that bacteria. And that is where we get the idea and the theory that we could program these bacteriophages to target those specific bacteria that are antibiotic resistant. So usually when we think of bacteria infections, we're automatically thinking of treating them with an antibiotic. Now, usually for a severe infection, that is our only option. But due to many instances of antibiotic misuses, we're seeing an absolute surplus of superbacteria that are immune or resistant to our most powerful antibiotics. So if this trend follows suit, we're going to come to an instance where there's going to be a bacteria that's immune to every major antibiotic that we have. So this is where the study and advancement in clinical trials of the bacteriophages come in. We could save thousands of lives and start winning the war on bacteria if we use these bacteriophages that are available for us. Sadly, there are many factors putting a pause on this kind of advancement. Sadly, bacteriophages are not immune to their destruction. CRISPR-Cas9 was the first instance of bacteria defending themselves against the bacteriophages. The bacteria basically learns that phage by taking a part of its DNA and storing it, so when it attacks again, the bacteria can prevent that phage's invasion. The human body's immune system is another one that can off the phages. Through phagocytosis, actually, the immune cells just engulf the phage and destroy them. So, like every treatment alternative that is out there are many weighing pros and cons for them. To be able to fully explore and learn all that these phages are capable of, there needs to be more funding and dedicated clinical trials to find their effectiveness. Sadly, it's not something that big pharma can profit from, really. So some independent company would have to make that happen. Knowing regulations for the safety of the research and also making sure legally that they are covered would definitely take some time to go through and enact. If that all happens somewhere in our lifetime, we would be able to save thousands of lives, just like the case study we discussed on Dr. Sean Patterson. His infection was successfully treated with bacteriophage therapy, and there's so many more patients that would benefit from this kind of treatment availability. Despite all the potential that bacteriophages have in this war on the superbacteria, we must overcome the regulatory and practical challenges to fully harness their medical capabilities and save countless lives in our future.